Well, good morning, everyone. This is EM-X for May 2024, and we'd like to welcome everyone to our session. And we got two wonderful speakers again, as we have in the past. So this will be the close of the American academic year. So this will be our last one, and we'll start again in October in the new academic year. So we are very pleased to have two speakers from Europe this time. And I will pass the baton over to my friend and colleague, Professor Hua Chu, who will introduce the first speaker. Hua.
Okay, good morning, good afternoon to everyone who come to these symposiums. It's my delighted pleasure to introduce our first speaker, Dr. Michael Wrench, from the Rosalind Franklin Institute in UK. And Michael started his experience in cryo-electron tomography at Oxford, where he got his training in structural biology. And so at that time, he already began to look into the possibility of integrating super resolution light microscopy and focus ion beam mirroring, and then also electron cryotomography. And then he moved to Max Planck Institute for Molecular Physiology in Germany, where he got a fellowship, and he even excelled this technology. And in 2021, he joined as a group leader in the Franklin Institute, where now he's heading a really vibrant and very productive group in pioneering the work in situ structural biologies. So it's my great pleasure to have Michael here today to share with some of his latest development in this highly demand technology for molecular and cellular biology. Michael, welcome.
Thank you. Thank you, Juan. Thank you, Bob, for inviting me. And I'm just going to share my screen. And hopefully you can see my slides. All good?
Yeah,
all
good.
No, really excited to be able to present today. So thank you for the opportunity, Bob and Juan. And what I'm going to talk to everyone today about is sort of our adventures in using cryo-electron tomography, but also crucially plasma focus ion beam milling and scan electron microscopy. And that's really geared towards, using this for in situ structural biology approaches and seeing where we can try and push the limits a little bit. And so kind of the dream in many respects for me and maybe for others is, the ability one day to be able to span the entire resolution spectrum. So, can we image molecules down on that atomic scale, but also in a pipeline where all of those factors feeding into those observations are preserved in the most native context. And obviously, this requires various different techniques, from hard X-ray micro-CT, which can image whole organs, for example, down to electron cryotomography, where you can image the molecules. But we obviously need tools. We need to build new modalities to be able to cross that scale. And so my group's really focusing around this green box here, sort of serial focused ion beam milling, scan electron microscopy, and electron cryotomography. But it's important to have that context, because if we're going to preserve the native organization of things in these in cells and tissues, we need to think a little bit more laterally about how we develop our workflows. And kind of part of that question is to build a complexity approach. So, many of you out there might look at cells, for example, with electron microscopy and look at the molecules in those. I like to say that's easy. Many of you probably don't think it's too easy, but what's important to note is that those cells, we don't exist as monolayers on electron microscopy grids. So building relevant biology and complexity is really important, but that comes at the cost of ease. And so fundamentally, what we want to do is marry the big with the small. So can we, natively image vitrified tissues, in this case, cardiac, some sort of heart tissue, which has been high pressure frozen and the water preserved, and one day target that specifically to look at the molecular structures that are inside of some of those biological structures. So on the right, you can see some work that I published a few years ago with Stefan Ranzer and Eric Wang, where we looked at the structure of myofibrils isolated from the psoas muscle of the mouse. And you can really see here the high resolution details, especially in this case, the purple protein nebulin, which up until that point, the structure hadn't been determined because it's very difficult to reconstitute that in vitro. So by looking in the tissues and using technologies to image those tissues, we can see some of those more native things that maybe we can't look at using classical biochemistry, for example. And so this is how we work. These are the two kind of modes that we predominantly use. So you have a dual beam FibSim, which many of you might be familiar with, where you use a focused ion beam to essentially ablate material on the grid. And typically we work at liquid nitrogen temperatures because we're working with natively preserved cells and tissues where we're trying to stop water from expanding, from becoming crystalline. And on the right-hand side, you can see a TEM. So we normally would fib, make a small lamella, as so-called, where you can see that you have a very thin region of the cell. And you might see on the right and bottom right, lots of little sputters, sort of, you see little speckles on that. Well, that's indicative of this imperfect freezing that sometimes we get, which is also a really important factor to think of when we're looking at structural biology approaches. And importantly, I haven't mentioned, but there's a scanning electron microscope on this dual beam instrument. So that means that we can actually image the material as we ablate it. At the Franklin, at the institute that I work at, we have several different microscopes. So we have a Helios hydroplasma fib, which has lift out, so we can use a cryocooled needle to extract bits of tissue, for example. And this has a high resolution scanning electron microscope, which allows us to do low kV imaging. And that's very important when you have biological material which has only carbon, oxygen, nitrogen, for example. So there's a weak interaction with the electrons and the sample. We have a prototype Arctis, which is the sort of latest development that we worked with Thermo Fisher on, where samples can be loaded 12 at a time in a robotic autoloader, so to speak, where you can essentially have long operation times at cryogenic temperatures. You can use different gases to mill your samples, and we also have a fluorescence module. And then we also have a Titan Cryos G4. In this case, we have a ±90 stage, which allows us to do some sort of different geometries, for example. So part of the talk title is about plasma ion beam milling. And so why plasma? Gallium is perfectly fine at low currents to mill very thin samples. And this has been demonstrated since 2007 by Mike Marco. Well, part of the reason why we want to use plasma is because of this blue circle here. As we go to very high currents, gallium becomes very divergent because of the nature of the optics and the extraction of the ions from the tip. Whereas at low current, gallium is has a very small probe size. So the idea was, can we use plasma to essentially have very high extractions, very high milling rates, but also try and preserve some of that ability to mill very thin material. So you can see by on this plot here, the probe size for plasma ion beams, in this case xenon, is much larger than gallium by a factor of around 5. So can we actually sort of use these ion beams? So part of the nature of the two ion sources, so LMIS, which is gallium ion sources predominantly because they have a low melting temperature and they're relatively stable. These are a point source, so you're talking 50 nanometers or so. And that means you can focus your ions to a very fine point, and you can get a very fine starting point for focusing your beam through the ion column. In contrast, when you have a plasma ion beam, you're using essentially an aperture, which is an area source. So you have a much larger point at which you're extracting ions, and that's why you essentially have the different coherence and beam shape. But obviously, the more you extract from that tip, the more these ions which have charge will essentially repel one another, and that's why when you try to extract very high ion currents, you get a divergent beam in the case of gallium. And for inductively coupled plasma ion sources, generally what you do is you have a gas inlet, and you essentially have an extraction electrode, and you pass an RF pulse across the gas to generate the plasma and start to generate a helical acceleration, which then comes through the gap. And I like this picture on the right because this is essentially a plasma ion. This is essentially a welding torch that you can use, and this is produced using an inductively coupled plasma torch. So essentially what's happening at the gun is happening on the right hand side, just in a more sort of sensible way. So a bit of the workflow. So part of this story is what we wanted to do is look at how we can use plasma to generate surfaces. So obviously, if we're interested in generating lamella at the end, we want to know how plasma ion beams behave when we're essentially polishing and producing flat surfaces. And flat surfaces are imageable using the scanning electron microscope. So as you mill, you can image, and you can image after milling several times in a sort of iterative fashion in a so-called destructive tomography. And part of this is important because what we want to know is which ion behaves better than the other. So if we have access to argon, xenon, nitrogen, oxygen. So we started doing some serial FibSem experiments, which you can see here. So on the left, you can see some bacteria. These are all vitrified, so we've either plunge frozen them or high pressure frozen them to try and preserve the native water content. In the middle, you can see the cardiac muscle I showed earlier, and on the right, you can see brain tissue. And what we really want to understand is what's the curtaining propensity. So obviously, different parts of samples mill at different rates if they've got a different content. And this will be what we want to understand is whether the different plasma ion sources had different propensities for this charge to build up. Curtaining. Because what this curtaining is essentially a topological profile. You know, how flat that sample is will be determined by how many differences in the curtaining profile that we get. We want to understand sort of what the resolution are and what information we can use practically. And so Maud was very determined. She wanted to understand which currents we should use for serial block tracer imaging like this. You know, what's the impact of automation? What gas produces the least amount of curtaining? And can we determine a metric that can explore this, for example, resolution? So you can see here a panel of different images acquired where you've basically milled a ramp, and then we've imaged the ramp and just tried to determine the curtaining. And in the bottom right, you can see nitrogen at high currents is a really large amount of curtaining. And so what Maud did was to essentially try and work out a score to assess this. So here, what you do is you do a Fourier transform of the surface, and then you look for the layer lines, and then you segment those layer lines out and remove the rest. And then what you essentially have is a plot of all the lines that's on the surface of that face. And we can then plot a curtaining score based on the number of lines, and then plot that as a function of current to see what propensity different gases have at different currents, for example. And you can hopefully see at the bottom left that xenon and argon are relatively similar at low current. So low current here is 0.1 to 0.2 nanoamp. But you can see as this goes to high currents, you have a much larger degree of curtaining. And so at the bottom left, we determined that argon is probably the best because this scale to the right is shallower. And so for making very flat surfaces, we are going with argon. Now, why is that? Well, on the top left, you can see the beam profiles where so in this case, you essentially do a spot burn in silicon. So you mill using a single pixel of your probe, and essentially then fill that up with something you can image, such as organoplatinum. And you can see that there's a relatively shallow full half maximum with gallium. On the right, you can see the case for argon. So you have this Gaussian, and the shape of this Gaussian determines how flat you can mill for a defined period of time. And this full width half maximum was essentially the smallest for argon at low currents. And so that's what we assumed was that this is the nature of the probes that is causing the curtaining to be the least when we have argon. So these are the kind of images you can get with serial FibSem. So this is essentially ablative imaging. You mill surface, and this is yeast, which has been plunge frozen. And you can see quite a lot of detail. So you can see nuclei, vacuoles. Sometimes you can make out mitochondria within these yeast. So actually, this is a very versatile tool for targeting, for example, if you don't have any fluorescence. We can do this on tissues. So here is a case where we can isolate a synapse, for example. In the red box, I've blown up the image here. So you can just about see the presynaptic phase where you see these various different vesicles. And on the postsynaptic phase, you can see this dark density. And so you can start to make out really complex biology and try to start to understand what's on the surface of your sample. And this is important, for example, if we're going for cryo-ET, where you want to make thin lamella, where you can image that on a larger scale or a higher resolution, sorry. This is just a sort of snap of that. And finally, just to kind of finish this off, what we tried to do was essentially understand if we could trace fluorescence through to the endpoint of cryo-TEM. So in this experiment, we're using sort of autofluorescent bacteria, and we can trace these bacteria through, and at the end find, in this case, in the orange box, using the SEM a septum, and trace that then onto the TEM at the endpoint, and you can see just about make out this lipid droplet at the bottom. And you might ask why these look quite different, the TEM overview and the SEM overview. And the answer is, well, with low KV and using a scanning electron microscope, you only penetrate 5 nanometers or so of the sample. And so the surface of this will look different to the projected image that you see in the panel below. So why plasma? So I've talked about them having the potential for high currents, but it's really important to maintain the fact that we can use the low currents to make lamella, because in the end, if we want to do structural biology, it's fine to have a very large hammer, but can we use that large hammer quite precisely? And so we set out to try and look to see if we could generate lamella for structural biology approaches. So here is Casper, Maud, and Tom. And as part of that, what we wanted to understand is the different trade-offs with the milling rate. So here's some very nice lamella, but what we also did was to try and measure how much material will move as a function of time and as a function of current. So, and also at different angles. And what we found is that xenon is the fastest for milling material. So you can see here, it's about twice as fast as the published values for gallium in water. And it's faster in nitrogen oxygen, but quite a bit, so about 4 times faster than argon. So argon is actually slower per unit charge than gallium, which was an interesting observation. And you might think, well, why would we use argon? Well, I just showed you, hopefully, that we can produce flatter lamella with argon. So we were using that case. So we were really using the flat aspect. We were being careful in this experiment. We didn't want to move fast, but obviously there is room at the top for xenon to try and increase the speed of this milling rate. We could do this relatively successfully, especially in an automated fashion. So we could produce sort of 34 lamella in 24 hours. Success means that we didn't break them in the SEM case. And then we produced, we acquired 180 tilt series from half of these lamella to try and take that forward for structural biology. And part of the reason for the 50% number here is that actually maybe sometimes when you brute force an experiment like this, you don't have any regions of interest that are suitable. So you might hit the nucleus, or in some cases maybe the cell was slightly too thick, so it wasn't vitrified properly. So we worked with 50% of our lamella. These are the representative images that we get from the TEM imaging of those lamella. So these are slices through tomograms of different thicknesses. So you have 138 nanometers, where you can see mitochondria with various different densities inside. And importantly, outside in the cytoplasm, you can see these different dark densities. These are ribosomes. And you can see these dark densities at different thicknesses. I mean, on the right-hand side, it's starting to get a little bit difficult to see, but you can still make those out. And these are just some further examples of some further biology. So you can see ribosomes in the referent pellet reticular, nuclear pore complexes, RNA vaults, and COP. So various different retrograde transport vesicles. So so this is the different thicknesses of those. You can see the mean average is 250 nanometers. So we can get, in an automated fashion, lamella relatively reproducibly, and we could probably improve this thickness. In fact, I know we can, because since we published this work, we're actually a little bit better at producing thinner lamella now. But we still still generally don't get it below 150 on average. So what we did was to take out the ribosomes that I just mentioned, and then there's an approach called subvolume averaging, where you can take ribosomes from within the 3-dimensional volume that you reconstruct from your tilt series in the TEM acquisition, and you can then try and reinforce the signal to noise in the subvolumes by aligning them all to a common reference. And in this case, when we did that using 16,000 particles from our data, you can see on the right-hand side that the resolution in the 60th subunit of the ribosome is around 3.8 angstroms. So that's getting, well, that is basically Nyquist in our case, but that's pretty close to pseudo-atomic resolution. And you can see in some of the helices, there are various different side chains, especially the bulky ones that you can make out. But importantly, we were interested in understanding what the important, what the trade-off might be with ion beam milling on the sample surface. So what we did was to take our 16,000 particles and then essentially bin them out into 15 nanometer bins. So if we take the edges of the lamella we produce and then plot to normal to that edge, and then calculate that distance, take those particles within that distance, and then average those separately to one another, we can plot a metric called a B factor, which allows us to determine how many particles we need to reach a certain resolution. And we did this and showed that after about 45 nanometers, the B factors don't improve. The reason being that if you think of your particle quality as being a Gaussian, that's where you essentially get to the midpoint of that Gaussian, where your particles are essentially the most productive in terms of alignment. And that's been done. That work has been done subsequently by Bronner and Lucas with gallium, and our data sets have been validated by Friedrich Forster using a sort of template matching approach that they have in house, and they calculate that damage to be 40 nanometers or so. So there's about 40 nanometers of damage produced using an ion beam to produce a lamella for structural biology. And that's really important to note. The resolution metric that we're using here is a very structural biology centric approach, where what we're interested in is, can we use these particles to look at structural biology inside these lamella? And since then, Helena has been pushing this really hard. So she's essentially been using xenon to trench mill high pressure frozen samples. And in her case, she's basically taking bacteria and producing lawns of bacteria. And bacteria have lots of ribosomes in them. So we're interested to see what the difference might be with Helena's samples. And interestingly, so we did exactly the same thing. We started to take bins, and on the back, interestingly, in this case, you have a 60 nanoamp trench. So what Helena and Casper were doing was to basically make relatively high current trenches so that they could get these sort of platforms for making lamella. And so the back is exposed to this relatively high current. We could see ribosomes in this, for example. So here's a bacteria ribosomes, and again, we could get a reasonably high resolution structure. But we then wanted to see how that compares to argon. And you can see here that in this plot, essentially the distance that the particles become, essentially the same is around, again, 40 to 45 nanometers. So again, xenon and argon have a substantial amount of damage down to about 40 to 45 nanometers. And your best particles come in the middle, obviously beyond that point. In the fun thing with the 60 nanoamp trench milling is that you can see here, for example, at the back of the lamella, you see this very large, so this is about a micron. You see a micron of amorphization. Now, in the material sciences world, amorphization essentially when you essentially mill a crystal, what happens to the crystal? Does it stay crystalline, or does it become non-crystalline? And that's called, when it becomes non-crystalline, that's amorphization. We don't have the same kind of nomenclature for this experiment, but what we can see is on the right-hand side here, there's a bacterium with a double membrane. And after this point where we see these structures generated presumably by the ion beam, you have still what looks like vitrified ice, but no biological densities. So the assumption here is that there's something happening when the ion beam interacts with the sample, which changes the structure of the molecules within the ice. But we still don't really fully understand that process. Just important to put this into context, there's quite a bit of work out there since we published some of the original damage studies which show gallium ion beam damage, and this is just a few examples. So there's a lab, this is Martin Beck's lab, there's some work which I think just recently came out as a fully published article, and they show that there's damage up to 30 nanometers, and then you get usable particles beyond that, or at least better particles. There's a recent paper by a Chinese group which showed a damage there of up to 15 nanometers for 30 kb. And I think what's really important to show with this study is that when you reduce the acceleration voltage that you use for the ion beams, you essentially reduce the damage layer down to 30 nanometers. So there's a lot of potential for low voltage ion beam milling, but it's important to point out that as you go to lower accelerating voltages, your probe becomes much larger. And if your probe is larger and your full width at maximum extends, then you're going to have a greater problem getting flat lamella and milling consistently. So there would be work to do in sort of generating that automation. So just to kind of finish off and summarize, what can we do to mitigate ion beam damage? Well, if you accelerate an ion at something, you're probably going to cause secondary cascades. So there's always probably going to be some sort of damage, but can we reduce that as much as possible? Well, high helium ion beams have a very small probe size. They interact weakly. The counteraction to that is, well, if they interact weakly, they're not going to mill very quickly. So there might be some trade-offs one day. Neon ion beams are used quite commonly, I think, in the material science as well, and they have a potential for a lower degree of damage. But these all need to be tested, obviously. Low KV milling, so probe size increases with at lower accelerating voltages. How would that impact on automation? Can we have some sort of alignment offsets? And then one really important characteristic that we're trying to understand, and I don't really have time to go into today, is what are the impact of high currents for FIB milling workflows and lift out, for example. So these are the various trade-offs between gallium ion beams and plasma ion beams, and I always like to show this because we're not really about, saying something is better than the other is such a binary thing to say. But obviously gallium, if you really want to make thin lamellar, thin samples, it's a great ion beam to use. But it is limited because of the current range. And one really important aspect to consider, and one of the real reasons why plasma ion beams were generated in the first place, is the fact that you can have implantation onto the surface of the sample. So if you have an aluminium sample, for example, and you use a gallium ion beam, you can get alloys. How that affects biological specimens is really difficult to determine, especially under cryogenic temperatures. Maybe eels would be able to tell us, but that requires very high electron doses. And on the flip side, ion beams are great. They have a large range of accessible currents, but they do have large probe sizes compared to gallium at the same current. And so then if you want to get really thin lamella, you might have to work a bit harder or do something a bit more exotic. And I think the automation for plasma is getting much better. In-house we are doing regularly automated lamella production, but there's a much greater experience with gallium. And so with that, I'm going to finish. I think I'm on time. And just acknowledge the group members. So this is the group, and this is the Franklin core team. And I'm just going to point out Helena, Maud, Tom, Casper, Matthew, and Jim. And then the rest of the folks at the RFI have helped out. So especially Michelle and Neville, who helped us a lot with some of the processing steps. And then our colleagues at Diamond, Martins Reed, Thermo Fisher, and the MPI in Dortmund. And these are the collaborators that we work with currently. So with that, I'm happy to take any questions. Thank you.
Hey, great. Thank you very much, Michael, for this really exciting development that you take a really critical look of what are the variables to optimize the sample preparations. The talk is open to questions. If you have any questions, please type into the Q&A. And Michael, you could probably read faster than I do on the Q&A.
Let me just find the chat. There we go.
Okay.
And the technique slide, what is the blur stuff on the tilt series? I think I mentioned that. I think there's some view. So essentially, if your sample is incompletely vitrified, you get reflections coming into your tilt series, which can essentially cause these little speckles. I think that's what you're referring to. Is there an aperture damaging issue regarding the FIB? So if you use, so this question's about the apertures. So essentially to change current, you have different apertures in your aperture slip. And as you mill, you're essentially milling away your aperture. So, the higher current you use, the more often you have to replace your aperture. So I wouldn't call it an issue per se, but the more often you use high currents, the more often you have to replace your aperture strip. The question about secondary electrons, do these also contribute to damage of proteins near the surface of the lamella? Possibly yes, but the mechanism of damage between ions and electrons, I think, is different. And I think in the case of ions, you're essentially liberating molecules that are larger than single atoms. So that kind of strikes me to think that it's a little bit more like when you're doing TEM imaging and you have loss of, for example, glutamates. That would be the kind of damage that you would have. The number of electrons that you would have though might not be particularly high. So the relevance of that, I'm not sure. What is the absolute highest current achieved for lamellar generation with PFIBs without destroying samples? How to deal with the charging artifacts? We have milled lamella with the highest current possible using the apertures we have available, and you can make lamella, however, they are not vitreous. So I think that answers that. What is the typical thickness of the lamella produced by the various beams? We haven't done that experiment because essentially you would have to optimize your polishing steps, and generally speaking, to get thin lamella, we use a lower current at the end. And we haven't done a robust study between argon and xenon with the various protocols, so I don't have an answer there, sorry. Can you comment on laser beam milling? So yes, so that's used quite a lot in material sciences to remove sort of silicon wafers, these kind of things, which are very hard. I think the amount of energy per probe size for a laser, in this case, I think we're talking 10, 50 nanometer femtosecond lasers, I think is quite high. So I imagine it would be not vitreous. So yeah, I think that would require quite a bit of research to figure out how to use a laser for ion beam milling. Well, not ion beam milling, obviously, but as part of that workflow.
Okay, Bob, your question. Mm-hmm.
Yeah, thanks. Wonderful talk, Michael. Thanks. This is we've done some ion beam milling with gallium on some brain tissue, and it was hard work. Let's put it that way.
Yeah.
But in material science, we tend to protect the surface layer with e-beam evaporation and then followed by ion beam evaporation, and that of course is standard practice. And so I'm wondering if you do that in your field, or because you can do deposit carbon instead of a metal, which is also highly protective of the surface.
Yeah. So the general practice in this space is to use an organoplatinum layer. So you essentially have a source of organoplatinum which you essentially place onto the sample before milling. So basically, you're protecting your very soft sample with a hard top piece. And generally, that empirically, we work out what time the nozzle, how much of that is basically deposited on the surface, and then so you have like a micron or so of material on top of your sample to protect it. And so the idea there is that any kind of residuals from your Gaussian probe is protected because the front of that is milling slow compared to the bit that you're actually milling. So that's standard practice, but we haven't done any of the things that you mentioned other than using organoplatinum, really. Yeah.
So do you have that capability in your cryofib?
Yes. Yeah. It's very difficult to mill without that. We've tried it. We've done various experiments without organoplatinum. It's possible, but the curtaining is much worse. So cells are not just water, they're like amorphous. Basically, they have a very diverse pool of constituents, lipid droplets, mitochondria with various solutes, this kind of thing. And if you just mill up front without any sort of hard surface at the front, you get very large degrees of curtaining, for example, because you have differential ablation rates.
Mm-hmm.
So it's quite difficult to mill without. It is possible, but it's not very easy.
Curtaining, yeah.
Yeah.
So
yeah, that's why Maud was so interested in figuring that out, or at least looking at that question.
Thank you.
there's a question on the Q&A.
How long can you keep the lamella in the chamber after thinning without considerable contamination? This was a really big problem a few years ago with different ion beam devices where essentially you had quite high water content in the chamber, and high is obviously relative. We're talking 10 to the -6 vacuum, but there would be like 50, 60 nanometers of material deposited on your sample per hour, I think. In our microscopes, so our Helios Hydra, we can't measure the contamination rate in ours. We've done it. And at the moment, our Arctis is around 4 nanometers per hour. So how long you can keep it is obviously up to you. You can take a considered approach. But I would say after some hours keeping it without polishing, you're going to have some ice on top. Although you mentioned the backside of lamella has artifacts, do you have a general estimate for a 10 micron lamella? What's the ratio regarding? I'm not sure I understand that question, I'm sorry. Okay, so for the trench milling. It's about a micron. So if you use a 60 nanometer probe, you get about a micron of damaged sample. So for a 10 micron, you would have 9 microns of usable space. Do you get contamination on the samples when moving from the FibSim to the TEM? Yes, it's a big problem. We try and filter our nitrogen very heavily, but we still get contamination. And that really depends sometimes on the sample itself. So we work with tissues quite a lot, and sometimes they're quite insulating and they get quite static. So it can be a real problem. There are solutions out there, none of which are cheap. But yeah, this is a problem.
Okay, I have a couple of questions. Have you encountered the problem of devitrifications in your pretty big chunks of tissues?
Yes. So I have a student working on this. They when you're working at high currents, there's a problem with devitrification up to at around 500 nanoamps, I would say. We start to see something. We are basically characterizing this currently, so I can't go into too much detail about the amount. But like I said, it's very black and white with 2.5 microamps. You have some devitrification.
I see. So it's a thickness dependence or is it positional dependence in your sample?
And what was the second part? Sorry.
Or the positional dependence. In other words, some areas appear to be devitrified when you move another area seem to be okay.
So we have worked quite a lot on our high pressure freezing to a point now that we can do multiple areas of one sample and it's almost always vitrified. And what the dependence of what I just explained is essentially the high current of the ion beam. And you can see there is a distance dependence of that. The further away you get from the point of milling, then you just get vitreous-fied ice again. Yeah.
Nice. Okay. There's some more questions on the Q&A.
Can you switch between different ions during milling? Yes, if you like. If you want to, then yes. You can automate that, I'm sure. There will be an offset with the alignments that you might want to consider because when you go from, for example, xenon to argon, you'll have a different set of alignments to try and focus that probe. But you can change the ion beam at any point. It takes about 10 minutes. Following Wild's question, have you compared the gallium and plasma performance regarding the deviation of large tissue sample? Performance is an interesting word. We can make very large blocks of tissue using plasma. I think the largest we've done is 450 microns. We're currently working out what the usability is of that approach. So one important thing to note, for example, if you have a vibratome slice of brain, if you cut that with a knife and then high pressure freeze it, regardless of whether you can what you can mill, 30 microns of that material is dead cells because essentially you're cutting cells in half and then the necrosis will happen in that 30 microns either side. So there is really quite a bit of utility for plasma if you're trying to get to the central part of your sample, which is the least damaged, for example. But again, these are quite long questions to answer. So we haven't fully answered them yet.
Okay. So what is your experience in the ARTIS so far?
The ARTIS is very good at milling in an automated fashion. And it's good for targeting on the fly. We have reached a point now where we can, it's basically used every day. We had some teething problems. So it's important to note that we had a prototype. So it was very early days for the development of that instrument. We had some issues with lost samples at the beginning, for example, due to various software issues. But it's actually, for milling cells on grids, it's great. For milling tissues, there's some work to be done because of the geometry constraints that is associated with milling at an angle. So yeah, we use the ARTIS all the time, but generally we're using that for thinning the miller rather than using high currents for bulk milling, let's say.
So I have one more last question. All the example you showed are mammalian origins. Have you tried some plant cells or plant tissue?
No, we haven't. We haven't worked with plant cells at the moment. We have a scientist who was planning to work on plant cells, but plant tissues, but they haven't got that far yet.
I see. Okay, good. All right, that's definitely is a really stimulating lectures you gave, Michael, and we look forward actually seeing you in person sometime in California.
That'd be great. Yeah. Oh,
Okay.
good.
Okay, let's turn the program back to Bob.
Thank you, Wah. Thank you, Michael. Good to know that we're sharing problems here from the specimen preparation point of view, as well as the electron microscope itself. So it gives me a lot of pleasure to introduce our next speaker, Professor Damien Ayoyo from University of Paris. And I've known him for quite a number of years now and carrying out wonderful in situ experiments in the TEM. And I particularly remember him filling in at the very last minute for a plenary speaker opening the one of the sessions at the Lyon European microscopy meeting in 2016. And he gave a simply fabulous talk and followed his work ever since. Anyway, so it gives me very, very pleasure to ask Damien, who enthusiastically accepted our invitation to give a talk, and he's going to tell us about the new work which is going on in his lab in Paris and with collaborators. Damien, please.
Yes, thank you, Bob, for the introduction, and thank you for this time giving me more than 2 days to prepare my talk. It's a great opportunity, and I'm also very excited and proud to add my name to this fantastic list of invited speakers that you had in this symposium. So my group, do you see my screen?
Yes, it's good.
Yes. Okay, perfect. Let me see if I can switch. Yeah. Okay. My group is working on the fabrication and structural characterization of nanomaterials, mostly for catalytic and biomedical application. And at the same time, we are running a national user facility for electron microscopy, including this double-corrected TM that give us the opportunity to exploit the multifunctionalities of electron microscopy to establish the link between the structure and the property of many different nanomaterials. When you try to develop nanomaterials for specific applications, you realize that establishing the link between the structure and the property is not enough, because at the end, you need a constant feedback loops between the fabrication, structure, properties, and performance of your materials. And that's the reason why we wanted to study all those different aspects of material development within the TEM. And that's the reason why for more than 10 years now, we have been working with closed cell TEM holders that give the opportunity to study nanomaterials either in liquid and in gas. And these well-established technologies consist in observing encapsulating liquid and gas between two silicon nitride membrane and controlling the composition of the environment with a microfluidic system that goes through the TA molder, so you can inject any type of gas or liquid in at the observation window, and you can observe the dynamics of nanomaterials either in their formation media or in their application media. And what we have been doing with those tools for more than 10 years now is studying the nucleation of growth of mostly nanoparticles in liquid environment. And we are also studying their transformation and degradation process either under harsh gaseous environment or in biological environment. And today my talk will only focus on this nucleation and growth study. So I come with this very interesting question that I asked to many of my colleagues, expert in nanochemistry and in wet chemical synthesis. Can liquid cell TEM become a game changer in nanochemistry? Meaning, do liquid cell TEM change the way we design materials? And when you ask this question to experts in nanomaterial synthesis, they will tell you it's a fantastic tool to obtain mechanistic information, but it did not yet change the way we design materials. So with many other colleagues and experts in liquid cell TEM, we discuss about how we could change this, induce this change of paradigm. And we realize that what we need to do is we need to work we need to have more realistic liquid cell environments, meaning that we have to keep developing liquid cell technology to be as close as possible to real media. We need to reduce the electron beam effect to improve, and for that we need to improve the signal to noise ratio of our images. We need to have a more practical use of liquid cell EM, which implies that we have always to compare in situ experiment with bench scale synthesis. This is the best way to generate new ideas and design materials in different ways. And we need also to develop correlative approach, which combine electron microscopy, of course, but also X-ray methods and optical imaging and spectroscopy. And the goal of my talk today is to show you what we have done in the last 5 years to work on those different challenges. So liquid cell EM, when you talk about the nucleation and growth of nanomaterials, you have to realize that they are simultaneously affected by kinetics, as kinetic effect, meaning the growth speed, and thermodynamic effect, meaning the structural stability of your nanomaterials in a given environment. And it's actually very difficult to distinguish these two effects if you don't have the possibility to visualize nanomaterials at the right scale in liquid. And that's the reason why for decades now, liquid cell EM has been deeply used to understand the growth mechanisms of metallic and bimetallic nanoparticles of different composition and shape, including nanoplates, nanodendrites, nanostars, nanocubes, and so on and so forth. And actually, most of the strategies that are employed in colloidal chemistry to control the shape of nanomaterials have been investigated with liquid cell EM. But still, we have only mechanistic information. So what we are doing, when you study the growth of nanoparticles, usually you are using the radiolysis of your solvent by the electron beam. If you take the example of water, you see that you split the water molecule into the various chemical species and radicals. And among those chemical species, you have high reductive agents like aqueous electron or hydrogen radicals that can reduce your metallic precursor to the metallic state and induce the nucleation and growth. And what is known for a long time now, because radiolysis is a well-known process, is that the steady state concentration of those reducing agents depend on the electron dose rate, meaning that we can use the electron dose rate to control the growth speed that allows you to control your reaction kinetics and observe the effect of this reaction kinetics. So that's what has been done for a long time. Here, an example from my group. You have a gold precursor that is irradiated in water. You observe the growth of nanoparticles. Here you see what's going on when you are in STEM mode here. when you increase the magnification in STEM, you decrease the scanned area, so the scanned area, so you increase the dose rate, and you see that when you increase the magnification, you increase the growth speeds. And now you can observe what is the effect of increasing the growth speed. You see that when you are in low magnification, meaning low dose rate, meaning low growth speeds, you form faceted nanoparticles. When you increase the magnification, you increase the growth rate, and you start having those branched morphology. And if you go at very high magnification, you start having those very rough surfaces. So you can interpret this result this way. Here, you gave me the deposit, everything, the shape of the nanocrystals depends on the competition between the deposition speed and the surface diffusion speed. The ability of the crystal to reconstruct well-faceted morphologies. So when the growth speed is low, you are governed by thermodynamics, meaning that you give enough time to the system to reconstruct faceted morphology. When you increase the growth speed, you are governed by kinetics, and the surface diffusion is not fast enough to reconstruct your thermodynamically stable facets. And with this type of imaging, we have been able to determine that the formation of faceted nanoparticles, for the case of gold, require reducing the reaction rate below 3 atomic layers per second. So this was great. This was work we have done a long time ago. Now, I told you that the first challenge that we have to meet is we have to work on more realistic liquid cell environment. And what we have done more recently is studying the effect of temperature. And actually, temperature is a very tricky parameter because any atomic scale mechanisms that is involved in the nucleation and growth of nanomaterials is actually temperature dependent. In the solution, the reaction, solubility, and diffusion of metal precursor depends on temperature. On the nanoparticles, the nucleation, absorption, desorption, diffusion of metal atoms, and also large-scale mechanisms like Ostwald-Ripning and coalescence depends on temperature. And even the atomic structure, the thermodynamic equilibrium, is also temperature dependent. So you definitively need an in situ probe to distinguish between these different effects. And here we have used those recently developed temperature control liquid cells that have been developed by Protochip, in which you have a resistance that is embedded inside the silicon wafer that allows you to control the temperature of the whole liquid cell between 25 and 100 degrees. And if you want to know more about how those liquid cell works, please have a look at these video articles we have done some years ago. And here I show you an example where we work in very low dose experiment, and you observe the growth of gold nanoparticles at 25, 50, and 85 degrees. And you see that temperature has a drastic effect on the nucleation and growth process. So we try to quantify this. And first thing we have done is quantify the nanoparticle density. And you see that at 25 degrees, you have several hundred of nanoparticles in your observation area. At 50 degrees, only 300, and only 30 nanoparticles are formed at 85 degrees. And you realize that, in fact, the nanoparticle density decreases because the nucleation period is much shorter. So when you now look at the nucleation theory, this is this equation that gives you the nucleation rates as a function of the supersaturation and at different temperature. You see that our experiment is not in agreement with this nucleation theory because we expect to have a higher growth nucleation rate at higher temperature, which is not the case of our experiment. So now we have to think about what's happened with this parameter, the supersaturation. Supersaturation is in fact the ratio between the concentration of gold precursor in your solution and the equilibrium solubility of your solutions. And what is great is that we can quantify the gold concentration, the gold precursor concentration in the solution, because it is the same in all the experiments at the beginning. And we can quantify the total amount of gold that we have inside the nanoparticles. And what is inside the nanoparticles is not anymore in the solution. And you see that with the growth, you have more gold inside those few large nanoparticles than in the many nanoparticles that you form at 25 degrees. So yes, it seems that the gold concentration decreases faster at high temperature, So you're happy, meaning that your saturation decrease. But now when you look at those arrows that correspond to the end of the nucleation period that you see here, you realize that in fact you have much less gold nanoparticles that have been consumed at 85 degrees at the end of the nucleation period than at 25 degrees. So it doesn't match anymore. So now we have to think about, is the equilibrium solubility of the solution depends on temperature? And the answer is yes. You just have to do this very simple experiment with salt. You put salt in cold water, you have a lot of salt residue. If you are above the equilibrium solubility, you heat up your solution here, you see that you have much less residue because the solubility increases with temperature. And it's also the case with this chloric acid. So how can we quantify this change? To do that, we analyze the growth rate of the nanoparticles. So here what you see is the evolution of the radius of the nanoparticles as a function of time. And you see that the qubit radius of the growth trajectory vary linearly with time. And that's what you expect when you have a diffusion limited growth. And in that case, what is interesting is that the growth now depends only on this growth constant, K. And in this growth constant, we have the equilibrium solubility. So we can quantify the change in solubility of the solution with temperature. And we obtain these different values. You see that the solubility increased 22 times from 25 to 85 degrees. And now we can revisit the nucleation theory and interpret why and understand why the nucleation period is shortened when the temperature increases, simply because you have a change in the solubility of your solution. So we were very happy to be able to really bring new information in the nucleation theory, but the idea of observing is really to go beyond the LSW theory. So we also have studied during this experiment the Oswald-Ripning of nanoparticles that you can clearly observe when you go at high temperature, like at 85. And the common view of the Oswald-Ripning is the growth of large nanoparticles at the expense of this small one. So you expect the small nanoparticles to shrink and the large nanoparticle to grow. And when you observe what's going on inside the liquid cells, you realize that this is true. On average, many nanoparticles, if you take the size of the nanoparticles, the mean size of the nanoparticles that shrink, on average, it's smaller than the nanoparticles that grow, but more than 20% of the nanoparticles that shrink have a radius much over these 57 nanometers. So it tells us that there are other parameters that are involved in Oswald-Ripning. It goes beyond size effect. So recently, through the postdoc of Yuvna, Vina Vidani Ranmaren, we have been working in a graphene liquid cell to study the Oswald-Ripning mechanisms, and we have been able to obtain these. But it works. No, it's not working. Video is not working. Let me try something. Okay, video won't work. Sorry about that. It's too bad. Sorry, it was working before when we test the video. Do you still hear me? Yes.
Yes.
Okay.
Let me see your screen. Can you share the screen?
Ah, sorry. Yes. Let me see.
Thank you.
Okay, let me see that. Hold on. Let me run. Up. Where is Zoom? Can you see my screen now? Yes.
Yes.
Good.
Yes.
Sorry about that. So yes, the video is not running, but what you were supposed to see is the dissolution of these nanoparticles. And in fact, what we could see, in fact, here you observe the evolution of the size of the nanoparticles as a function of time. And what we have been able to demonstrate is that there is a directional, actually nanoparticles are constantly exchanging matter during growth. And in fact, through this video, we have been able to demonstrate that there are actually a directional atomic diffusion that is in fact driven by the position of the disruption site. And that's what you can see here. Nanoparticle 1 at the beginning, it decreases in size, while nanoparticles 3 increase because it's the disruption sites are in the southwest region of nanoparticle 1, feeding nanoparticle 3, and then you have nanoparticle 2 that lose materials from the south region, reaching nanoparticle 1 that increase in size, and then nanoparticle 1 completely disappear because the flux of atoms between nanoparticle 1 and nanoparticle 3 increase drastically. So in fact, if you want to understand the Ostwald-Riepning process and what particles will disappear during Ostwald-Riepning process, you should not take into account the intrinsic structural stability of a given nanoparticles, but rather the atomic flux between all the nanoparticles. So it also depends on their environment. The other things we have been able to show in this in this temperature control experiment is the possibility to visualize the whole nanophase diagram of nanoparticles. And this nanophase diagram really depends on this competition between deposition rate and diffusion rate. And we have been able to confirm a theory proposed by Professor Xia at Georgia Tech University, where he has shown that starting from a cubic palladium seed, depending on the growth speed, if you are dominated by the growth speed, so by the deposition speed, you grow dendritic nanoparticles. If you are dominated by the diffusion at the surface of the nanoparticles, you are forming well-stable 1-1-1 facets. And in the middle, in the intermediate regime, when you play with these two parameters, you are able to grow nanocubes. And what is interesting in our liquid cell TM experiment is that we can actually control the deposition speed, both with the dose rate and the temperature, but the diffusion of atom at the surface of the nanoparticles only depends on the temperature. So we have a way to play with these two parameters. And that's what we have done. You see that when you work in low dose rate, either at high temperature or low temperature, you are dominated by the diffusion speed. And you are forming nanoparticles that have mostly 1-1-1 surfaces, well-faceted nanoparticles. You have already seen that when you increase the dose rate at low temperature, you grow rough surfaces, dendritic nanoparticles with rough surfaces, because you are in between, you are dominated by the deposition speed. And when you work at high dose rate but high temperature, you reach this intermediate regime when you start forming those nanocubes. So I show you that playing with temperature gives you another possibility to explore the nanophase diagram of nanomaterials and better understand their formation processes. Now, the second challenge I wanted to discuss is the necessity to work at much lower dose rates, because we want to minimize the electron beam effect. And if you want to meet this challenge, you need to improve the signal-to-noise ratio of images. And actually, when you work on those videos, even if you work with gold nanoparticles, treating video processing is actually a nightmare, because even if you work with gold, the signal-to-noise ratio of those nanoparticles is very poor, between 1 and 2, because you cross this thick layer of liquids. And additionally, your background is not at all homogeneous, because you have a lot of particles that grow on the opposite membrane. that are out of focus and that drastically contribute to the inhomogeneous background of your images. So to solve this issue, we try to work with a deep learning denoising approach that has been developed by one of our PhD students, Adrien Moncomble. And Adrien has developed an approach which consists in training a neuronal network with kinematic STEM-HDF simulations that provide an image without noise, that takes only into account the size, the shape, and the orientation of the various objects that you can create. So it's only a thickness map, and then you calculate the intensity of your nanoparticles through this very simple equation. And then you give also to the network a noisy image that take into account the spread of the beam due to the liquid, the high level of noise due to the liquid cell sample, and all the out of focus object that you can add to your image only by changing the size of the beam that can image those objects. And you see that the image that we simulate here are very similar to our experimental image. And we can give to the neuronal network these noiseless images that allows the network to reconstruct extremely well-defined images of our nanoparticles with a signal-to-noise ratio up to 10. So now that's provide a lot of new possibilities. Here I show you a comparison between an original image, what you can obtain when you applied conventional filter strategies, what you can obtain with noise to void deep learning approach, which is a neuronal network that is trained only with noisy image. And this is our approach using a UNet and a specially designed UNets developed by one of our collaborators at EFPEN. And you see that in terms of image quality and in terms of signal to noise ratio, our approach is definitively better than other conventional method to denoise liquid cell TM image. And what is very interesting here that it also works on videos. So it gives us the possibility to perform in situ STEM experiment at much lower dose rate, because now we can improve the contrast of nanoparticles after the experiment. It's very easily adaptable to other particles and beam-sensitive materials. It takes only a few hours to train the networks with a new set of samples. And it's also extremely fast. Once you have trained your network, it's actually extremely fast. So we can now consider this on-the-fly to obtain on-the-fly denoising to obtain this type of image directly on the microscope. So this will be out soon in the articles of Adrien, and all the code will be in open access to allow the community to benefit from this new denoising, deep learning assisted denoising methods. Now, the third challenge, remember, I told you that it's extremely important to be practical, okay? More practical use of liquid cell EM. Because if you want to fill the gap between in situ experiment and bench scale synthesis, you need to compare them. You always need to have the experiment run at the bench scale, at the Beaker scale, and compare your in situ observation. And I'd like to show you a collaborative work that we perform with the ETODIS laboratory. We work on biotemplated synthesis of gold nanoparticles on TIMV viruses. So biotemplated synthesis is performed to control the distribution of gold nanoparticles in 3 dimensions. And here the idea, my colleagues, they want to develop a SIRS probe. So the idea is to add a lot of gold nanoparticles on those viruses, because when you increase the density of nanoparticles, you increase the plasmonic interaction in between the nanoparticles, which open their plasmonic resonance in the biological windows. So the challenge they wanted to meet was very simple: how we can increase the density of gold nanoparticles at the surface of TMV. So that we have done the same experiment as before, except that we have the TMV viruses inside the liquid cells. And we have, from the mechanistic point of view, we have been able to confirm that the nanoparticles, they don't form in solution and then grab graft to the viruses. They really form on the viruses because there is an interaction, electrostatic interaction between the precursor and the surface of the virus. You have the reduction of your metal precursor, the growth, and the very rapid coalescence of the nanoparticles. Now, what's going on when you increase the dose rate? You increase the dose rate, you increase the concentration of reducing agent, you increase obviously the growth speed. But what is interesting is that when you increase the growth rate, you also increase at the very beginning of the experiment the density of nanoparticles. So you have a very high burst of nucleation at very high concentration of reducing agent. But then the density decreases because very rapidly we have those coalescence mechanisms. And when you compare the result or in situ observation with bench scale synthesis, so my colleagues, they have run the same experiment, except that they are using NABH4 as a reducing agent with different concentration, and they run different cycle of reduction, and we observe the result of the synthesis after each cycles. Same result, you see that you increase the concentration of reducing agent, you increase the growth speed, and also you increase the density of nanoparticles at the first cycles, and coalescence drastically decrease your density of particles. And from those observations, we have been able to design a new way to fabricate biotemplated gold-coated TMV virus by increasing the concentration of gold of reducing agent at the first cycles, and then using very low concentration of reducing agent during the growth phase. My colleagues have been able to fabricate those virus decorated with a very high density of nanoparticles that is up to 10 times higher than when you conventionally use the same concentration of reducing agent at each cycles. And that's what I mean when I say we need to be more practical. We need to bring concrete information to colloidal chemists to convince them that in situ information can change the way they are fabricating materials. And those nanomaterials, you see that from the optical point of view, they have this window, this absorption that is now very wide, and you have absorption in the infrared region, and those nanoparticles, those hybrid nanosystems, they are also very efficient for cells detection in solutions. So that's an ongoing work. Now, last challenge I wanted to discuss was the development of multi-scale and multimodal approach. Just to show you that it's actually extremely important to know if what's happened in our liquid cell, do we have the same process at the bulk scale? And I'm going to show you two examples, one case where we have the same process and one case where we don't have the same process. First experiment, we run the growth of silver at the surface of these bipyramidal gold seed. Very simple growth process. You have the bipyramidal gold seeds, you had the silver precursor, the ascorbic acid, and the CTAC. Here is what you get when you follow this simple reaction by Sachs. What those curves tells you is that you have the growth of anisotropic object with a constant width. And if you want to have a perfect fit on your Sachs curve, you need also to add in your model a lot of small spherical silver nanoparticles that grow together with the nanorods. And now if you follow what's going on with absorption spectroscopy, you see that first you have a blue shift and then a red shift. So now let's see what's going on if you reproduce exactly the same experiment in the, it's not running. I don't know why it's not running. Well, anyway, what you see, you can see here in the contour plot of the nanoparticles, you see that first we observe the growth of anisotropic object on the bipyramidal seeds. We also observe the growth of many spherical nanoparticles all around. So we were very happy. We have the same conclusion as with Sachs. And you see that, in fact, we have a transformation. The nucleation site of the nanoparticles at the beginning are the common base of the bipyramidal seeds, which has 100 edges. And in fact, you have the transformation of those edges into 100 facets to grow those nanocubes. And you see that the length of your nano object is not increasing at the beginning. You just change the shape of your nano object. And now if you run beam simulation to simulate the absorption spectra of a given nano object, and you add this layer and transforming the bipyramidal seed into a nanorod with a pentagonal symmetry, you understand why you have this blue shift. And then once you have the nanorod shapes, you have the size increase of these nanorods, and the longer the nanorods, increasing the size of the nanorods induce the redshift. So you see that the possibility to visualize the intermediate structure and the reaction dynamics allows sometimes it's solo explaining well what's going on at the bulk scale and in the beaker of our colleagues. Now this video won't run either. This it's all something that was interesting was what was driving the anisotropic growth. Many of if I come back here, you see that many of my colleagues, they saw that it was due to the CTAC, which is known to stabilize given facets of metallic nanoparticles. But in fact, in this experiment, we have been able to run exactly the same growth experiment, but without the ascorbic acid, which is known to be only the reducing agent. And it turned out that when you remove the ascorbic acid, you see that you start having an isotropic growth, where you have a growth of both the length and the width of your nanoparticles. So it gives also the possibility to reveal the role of each chemical agent during the synthesis. And here the role of ascorbic acid, it's a source of electron to reduce your metal precursor, but it's also a shape directing agent that drives the anisotropic growth of silver nanorods. So for this experiment, we were very happy. The process observed in the liquid cell and at the bulk scale by Sachs and absorption spectroscopy worked well. Now I'm going to show you another experiment with the same type of object, where we try to study the formation of supercrystal by depletion forces using the same silver nanorods and other nanoparticles which were gold nanoprisms. So depletion forces, it's an attractive force that can be controlled by the concentration of and the size of micelles inside your reaction media. And when you see that when you increase the concentration of CTAC, this year we are using, my colleagues, they are using CTACs, you increase the concentration, you start to see this pattern on your Sachs curves that describe the formation, in the case of silver nanorods, of 2D hexagonal lattice, and you can confirm that by doing XC2 SEM. And when you work with the nanoprisms, the nanoprism form a 3D interlock hexagonal lattice, but also confirmed by XC2 SEM observation. Now what's going on when you run the same experiment inside the liquid cell? We see that we form also supercrystals, except that the affinity of the nanoparticles for the membrane prevents them to stack in the direction perpendicular to the membrane. So we end up with 1D strip of silver nanorods instead of the 2D hexagonal lattice, and a 2D interlock hexagonal lattice for the gold nanoprisms. In fact, you can observe the interlock lattice along different directions, but the ability to stack perpendicularly to the membrane is limited to 1 or 2 layers. So at the end, what we have in the liquid cell, we have nanocrystal with the same structure, the same lattice parameter, but with a much lower dimensionality. And at the end, my colleagues were very happy because they say that we have now the opportunity, by adding a membrane, to change the structure of our supercrystal. So it's a way to control the structure of supercrystals, but it's also crucially important to know that you don't necessarily always have the same result inside your liquid cell and at the bulk scale. And we wanted to understand why. And we have studied the degradation kinetics of those nano objects through the electron beam. And you see that over time, you have a loss of the crystallinity of the supercrystals. And they all have the kinetics of degradation is always the same. First, you have a plateau, and then a drastic drop in the number of nanoparticles that you have inside your nanocrystals. And we fit those curves with a kinetic model that account for the desorption of the nanoparticles. And what is interesting with these models is that you have in this A term here, it depends on the adsorption energy. So all the force that stick the nanoparticles on the membrane, and a term that depends on the cohesion energy, that all the force that holds together the supercrystals. And you see that when you change the dose rate, the cohesion terms is not varying, whereas the adhesion term is drastically changing, meaning that, so these results indicate that we are not degrading the C-tact, we are changing the affinity of the nanoparticles for the membrane. And it is well known that when you irradiate the membrane, you change the charge of this membrane, and by electrification process, you can reach a status where your nanoparticles have the same charge than the membrane, which favor its expulsion. So this kinetic study prove that the structural properties of those nanocrystals is affected by membrane charging and the affinity of the nanoparticles for the membrane. So I come to my conclusion, and I come back to my original question: can liquid cell TEM become a game changer in nanochemistry? Today, my answer will be it's on its way. And I'm pretty sure that if we keep working on the different challenge and aspect of this game, we will convince more and more colloidal chemists that liquid cell TEM can be a tool not only to provide mechanistic information, but a tool to provide information to design, to change the way we design nanomaterials. And with this, I'd like to thank all my coworkers in my group and beyond. And I thank you very much for your attention. Sorry for the video that did not work. And just a little advertisement. Next year in May, we will organize the 6th edition of the Quantitative Electron Microscopy School. And it's a great event that we can have more than 100 participants. So if you or your student wants to enjoy the beautiful south of France and spend 2 weeks learning many aspects of imaging and spectroscopy techniques, including in situ TEM, please save the date. And again, thank you for your attention.
Great. Thank you very much, Damien, and certainly learning about these things in the south of France. So what a combination. So after there should be some the audience should ask questions most easily through Q&A, please. But I have a couple of questions, of course, about the use of the electron beam and compared to the bulk experiments. So I'm wondering if maybe at some point we need to have bulk experiments which involve an electron beam as well, as an extra stimulant since we can perfect particular growth mechanisms using the electron beam. Maybe it should be incorporated into a bulk process as well.
For sure, it's actually a very interesting strategy, and there are many labs in the US. I think Notre Dame University is expert in radiolysis synthesis of nanomaterials at the bulk scale. In France, we have a very good group in this in radiation chemistry also at the University of Orsay. The main issue you will have with this strategy is that the dose rate level that we reach in the liquid cell TM is a few decades higher than what you can obtain with other type of irradiation at the bulk scale. So yeah, there will be always a difference in the dose rate. But yes, that's the definitely that's the closest bulk scale experiment that you can run to be as close as possible to what's going on in the liquid cell TM experiments.
Right, because sometimes the electron beam can help. So,
oh yeah, yeah, definitely.
Anyway, the other thing I had is that, you know, a lot of our colleagues here, they do make nanoparticles and often by liquid reactions, and the shapes and so on are controlled very highly by the surfactants which are used.
Mm-hmm.
And I'm wondering, and often, of course, when you've got a liquid solution, you can throw in the surfactants at the end or at any point during the reaction, which can control the shapes and the sizes and so on.
Yes.
And I'm wondering to what effect you're studying surfactants.
Some years ago, it is definitely possible to study the phase blocking methods. Okay, it's a well-known strategy, and you can definitively study the role of a given surfactant in the liquid cell. And it's actually I think the main strength of observing at this scale is really the possibility to study the role of each component in your synthesis. And something we have done some years ago was to study the growth of nanostars. And it was driven only by one surfactant, which was dimethylamine. And just by injecting the dimethylamine during the growth experiment, it changed the way it changed completely the growth process, the growth mechanisms, and you transform icosahedral nanoparticles into nanostars. So yes, the answer is yes, and there has been many example of liquid cell TM studies studying this type of process.
And any application of eels yet to study those effects as well on the surface plasmons?
The problem is that the energy resolution of the beam is drastically affected by the liquid environment. So if you want to do eels, I suggest to work in graphene liquid cell, because you will have less effect of the membrane and you can have much thinner liquid cell. There are some work out from Professor Meyer has done some work in using eels in liquid cell, and actually plasmon is the region of the energy loss spectra where you have the more application. So yes, it might be possible with graphene liquid cell to study plasmonic nanoparticles.
Good, thank you. If you go to Q&A, Kate Reedy
Yes.
has a couple of questions, please.
Okay. I wonder, have you
Kate.
ever thought of? I wonder, have you ever thought about surface treating the membrane to get more or less nucleation or tailor epitaxial growth? On a regular basis, we treat the silicon nitride membrane with oxygen plasma to turn them hydrophilic. So it's help for the sample preparation. Sometimes we want to see the effect of we want to see the effect of having an hydrophobic membrane. It's possible to load the sample even if you don't run this plasma process. But that's all we have done so far regarding the treatment of the surface. But I know that in biology there are also strategy to trap the protein or a virus at the surface of membrane. So some strategy exists. And the second question is, I suppose that the mechanism of nucleation on the membrane in a low volume of liquid is a major difference to experiment carried out in the bench scale, on the bench, and are there a way to mitigate this? The thing is that yes, definitively the membrane has an effect for sure. The fact that you have an interface, it become an heterogeneous nucleation. But usually we like to work on that's also where most of the nanoparticles are formed, because that's where you have the more radiolytic effect, because the membrane generates a lot of secondary electrons, and secondary electron contributes a lot to radiolysis. Now, if we wanted really to study the growth in liquid, I guess it's possible, because people have seen, for example, that there are some particles that diffuse in the liquid with normal Brownian diffusion. So there are some bulk scale process that are in the that happen in the liquid cell. The key is to observe them, and obviously it's much easier to observe a particle that doesn't move rather than a particle that moves. So yes, maybe there are ways to there are particles that form in the liquids. The key challenge will be to catch these specific nanoparticles.
Thank you.
I think there is also one attendee raised the hand. So, hi,
yes.
Karen, can you hear me?
So, thank you. What do you have any comments, or Michael?
I don't, but Karen Oakley has a questions. Let's see if we can unmute her.
Yeah, she's really the panelist, so she can unmute herself and talk.
Oh. All right. She put her hand down. Okay. I have a question for Damien. So when I look at your TMV particles, it looks like some of them are aggregating together.
Yes.
Why is that the case?
It's a good question. Maybe we did not. Actually, it's probably a membrane effect. It's probably something that happens on the membrane, because I remember that when you do ex situ TEM, we also found some particles that aggregates. But it is true that they were always like connected tip to tip when they were in the liquid cell. So probably just like the formation of supercrystals, the spatial distribution of the TMV viruses is also affected by the affinity with the membrane or the confined space.
Okay.
And
have you tried other types of nanoparticles than TMV?
Yes, you can. We have run some experiment for. We have studied the mineralization of manganese at the surface of bacteria, using exactly the same type of experiment for another. It was really a geochemical study. My colleagues, they want to observe the effect of the exopolymer that are produced by different type of bacteria, and they want to study what is the effect of those exopolymer on the mineralization of manganese. But the answer is yes, you can study other type of biological object. You don't do live imaging, okay? But you can use the capsid or the membrane of a biological object as a substrate for growth experiments. Yes, that can be done very easily.
I see. In that experiment, do you see the manganese ions to aggregate on the surface?
Yes, and it's a Science Advance paper of 2001, of 2020. Sorry, I'm not that old. 2020, the first author is Kwasno, Thais Tuquasno, and we show that depending on the strand of the bacteria, the one that produce a lot of EPS and the one that does not produce a lot of EPS, we have completely different nucleation and growth process of the manganese at their surface.
I see. How do you know they are manganese in the studies?
Oh, they are manganese because we put manganese salts inside the liquid cell.
I see.
We intentionally had the manganese.
I see. Maybe you can do a use experiment to show that is the case or not, perhaps.
What?
To do a
Yields. Oh yeah. Oh yeah. I did not show it, but what we do every day is EDX. EDX is much.
Yeah. I think Kate has another question on the Q&A, Damian.
He seemed to be frozen.
Damian, are you there?
Yeah. No, I'm here. You can hear me?
Yes. Have you got Kate's question on the Q&A?
Yes. Sorry about disconnection. I also really enjoy the focus on correlative in situ analysis. Have you developed ways to use the same liquid cell tip in X-ray beamlines, etc., or is it too low volume? Yes, the answer is yes. That's actually the project of one of my recently enrolled CNRS colleagues, Nathalie Ortiz. Her project is really to develop liquid cells that will be compatible with STIXM, liquid cell TEM, and optical methods. So yeah, it's on its way again. And yes, that's definitively the future, one of the future challenge that we have to meet in the field, because we can see actually that if you don't have like another type of experiment at different scale or using a different technique, you don't know if what you observe is an artifact or it's always the truth, but is it an artifact or is it meaningful information? It's crucially important to have correlative approach.
Right. That's the critical. That's a critical statement. One last question, which for me, the perennial question: what KV is best for this work?
It really depends on what you want to do. The lowering your liquid cell, your high voltage will increase radiolysis effect. So usually I recommend to use high voltage, but if you work with graphene liquid cell, then you could start having some knock-on damage on your graphene. So people recommend to work at 80 kV. So if you want an easy answer, I will say in silicon nitride, work in at 200 kV and work at 80 kV with graphene liquid cell.
Yeah, I always tell people to try it and see see which works best.
Yeah.
Great. I think we should wrap up now. Ward, do you have final comments, and then I will close out the session?
No, I don't. I think there has been a wonderful morning to hear two wonderful lectures. And I definitely learned a lot about the technologies, and we look forward to meeting you in person. Maybe in southern France, it's a fantastic idea. I like that.
You're very welcome.
Right, and you'll learn a lot about cryo there as well from Wa and his colleagues. Great. So firstly, I'd like to thank Michael and Damian for a fantastic way to finish out our academic year. Thank you both. Terrific talks, very stimulating. And then I'd like to wish everyone a great summer. And maybe we'll meet, if not in the south of France, and over conferences over the summer. And we'll look forward to seeing everyone again in the autumn, likely the first Monday of October. So please mark that on your calendars. Of course, I'd like to thank Yi and Jane for fantastic organization here. Yi, you've been terrific this whole year. And thanks again for coordinating this.
Thank you.
Yes, thank you, Yi. Thank you, Yi, for your time.
Well, Damian, Michael, thank you so much.
Right.
And
Yes.
See everyone in the future. Bye-bye.
Sure. Thank you very much, Bob. Thank you.
Bye. Thank you.
Bye.